Hello and welcome to another Bio 111 lecture. Today's topic is DNA structure and the history of DNA. Before we start, let's take a look back at what we talked about last time. So last time we talked about biomolecules, and specifically we talked about the building blocks and how the building blocks make up the larger molecules, monomers versus polymers. Specifically, we talked about amino acids that became proteins, fatty acids that become fats, monosaccharides that become polysaccharides or sugars, and then the nucleotides that make the DNA and RNA strands. And this is something that we'll discuss in more detail today. All of these reactions, not all of them, but many of these reactions are a specific type of reaction called the condensation or dehydration reaction. So many of those reactions where building blocks form polymers go through this condensation or dehydration reaction. And we talked about the names of the newly formed bonds in all of these classes of biomolecules. So we talked about the peptide bonds for the proteins. We talked about ester bonds for the fats, glycosidic bonds for carbohydrates, and phosphodiester bonds for the DNA. And then we talked about polarity and how electronegativity plays a big part in determining whether or not a molecule is polar. Polar or nonpolar also determines whether a molecule is hydrophilic or hydrophobic. And we talked about cell membranes and phospholipids that make up these cell membranes and how polarity in those molecules is very important for creating a film or a membrane that can allow a cell to have a border within an aqueous environment. Okay, so today we want to talk about DNA. And we want to go back a little bit in history and see how DNA was found to be the molecule that is this substance of inheritance. So let's take a look at an experiment that was done in 1928 by a fellow called Griffith. So what Griffith did is he wanted to determine what happens. He really was a bacteriologist. He was interested in streptococcus. He was interested in the kinds of bacteria that make people and animals sick. And so he was working with different types of bacteria, virulent and non-virulent strains. And so they were sort of the same species, if you want to call it, the same isolate, but they were different. So they were called different strains. And the way that they were different from each other was that one set of these bacteria, one type of bacteria, was virulent, causing a pathogenic reaction, a disease. And then the non-virulent bacteria, which didn't have any effect. And so they also were different in shape. So he could look at them under the microscope and you could see that one was rough and the other one was smooth on the outside. So he could also determine them phenotypically, it's called, by their appearance, in this case through the microscope. Okay, so he did these 4 types of experiments where he injected mice with the virulent bacteria, and that caused pneumonia, and the mouse would die after 2 days. He also used the non-virulent, the non-virulent strain, and when he injected mice with that, the mice remained healthy. And then when he killed the virulent bacteria by boiling them first and then injecting them into the mouse, he was basically working with a dead set of bacteria that he injected, and in that case, the mouse remained healthy. So killed bacteria didn't cause the disease. But then, to his great surprise, when he killed the virulent bacteria first, so this, and then he mixed them with living non-virulent bacteria that previously had not caused any harm to the mouse, then the mouse died. So that was puzzling to him. And the conclusion here was that if you look at this experiment where you inject the bacteria into the mouse and the mouse dies, and they inject the non-virulent and the mouse lives, and you inject the killed bacteria and the mouse lives, and then you have the mix of those two where the non-virulent seems to somehow become virulent and kills the mouse. His conclusion was that some molecule in the dead virulent bacteria can be taken up by the live non-virulent bacteria. So something that through the mixing, was transferred from the dead bacteria to the living bacteria. And that gave them this new property that was the one of virulence. So this new property for the previously non-virulent bacteria, acquiring a property from a dead substance that had some sort of a molecule in it that was transferred to the living cells. And so he called this process of acquisition transformation. He transformed the bacteria with a substance that he didn't know what it was. It took quite a few years until it became clear what that was, what that substance was that killed the bacteria, that killed the mice in the bacteria, through the bacteria. And that was in 1944 when Avery and two other colleagues sort of repeated the experiment, but only parts of it. So let me walk you through what exactly they did. Think about that last experiment that Griffith did, where he mixed the bacteria together, the living non-virulent and the dead virulent bacteria. So that's basically what he did here. He had killed virulent bacteria, and eventually he would mix that with the non-virulent bacteria. And he did that in 4 different experiments. So this last set of experiment was basically done 4 times, but slightly differently. So each time he would add the virulent killed bacteria to a flask of non-virulent bacteria. But before he did that, he would either do nothing in this experiment. He would add something that's called RNase. That is an enzyme, an active protein. We'll talk more about enzymes later, but it's a substance that kills RNA or degrades RNA. So he added RNase, and that broke down any kind of RNA that was still in this substance here from the killed bacteria, but nothing else, only RNA. So everything that he put in here was now the whole thing minus RNA. And then he did the same thing, but he left out the RNase and added in protease instead. And protease, as you might guess from the name, is again an enzyme. Can already tell you all enzymes end in ASEA. So whenever you see ACE, that is an enzyme, a protein that has a certain activity, cellular activity. So this is a protease that he added that would degrade all protein in there. And then he added that to the non-virulent bacteria. Or he added an enzyme that was called DNase, and DNase breaks down all DNA in this mixture, in the substance of the virulent bacteria, the killed virulent bacteria. So first the enzymes or nothing added to to a subset of the bacteria, then that was added to the non-virulent bacteria. Then he would see what would happen to that bacterial mixture by injecting it into bacteria, into mice, for example. And what he found was that he only got non-virulent bacteria when he added DNase, suggesting that adding DNase to this dead mixture of cells and buffer and water and so forth, and DNA, of course, that the breaking down of DNA led to the non-virulent appearance of the non-virulent bacteria. But whenever he had DNA in the substance, here, here, and here, where he didn't degrade the DNA, where he degraded something else, he still, in the end, had transformation where the bacteria picked up the substance, the DNA, as he then found out, or as they, I should say, then found out, because it was 3 of them who published the results, and therefore suggesting that it must be DNA that is transferring this particular trait. So I wrote this down here for you. You can read it. You can read this. I'm not going to read this all to you, but you can stop the lecture, take a look at it and read it. But the upshot, of course, is that only DNA has the ability to transform the bacteria. Okay, so that is a little bit of history of how people knew that DNA was in fact the substance that was transferring capabilities, cellular capabilities, from one generation to the next generation. Previously, people thought it could be RNA, it could be protein, it could be something else. But this experiment showed quite nicely that it most likely was DNA. This is in 44, so this was still quite a while before the structure of DNA was known. So the structure of the double helix that I've already shown you in the previous lecture. People didn't know that, but they did. They were able to isolate DNA. It was just sort of a globby substance that they could isolate, and so they knew that that was there. They just didn't really know the molecular structure of it. But that was in 1944. Okay, let's forward in time quite a bit, and I'm going to tell you something about what's called the central dogma. So this is an important dogma, a rule, if you will, that was derived from many years of experimentation. And this is what we now have known for quite a while, that the information flow, the heritable information flow from one cell to the next cell goes through these steps. So it starts with DNA, as was found out in the previous two experiments that I showed you, that then a process called transcription occurs, in which the DNA is transcribed into an intermediary compound. So this intermediary compound is called RNA. And specifically, it's called mRNA or messenger RNA. There are other types of RNA that are also made by transcription, but they don't transfer the message to the last step, which is the protein. So we'll talk about the other types of RNAs later, but right now, this is the messenger RNA, the mRNA. So mRNA is made by unzipping the DNA and then producing a single strand of RNA, mRNA in this case, that then, here you see, is translated into the protein, and these are the proteins. So the proteins then can be any kind of proteins. That can be structural proteins that give the organism certain shape, for example, or it could be an enzyme that has an activity, for example, a metabolic activity where sugars are broken down and energy is made, or things like that. So those are enzymes. All enzymes are proteins, but not all proteins are enzymes. Right? Structural proteins, for example, are not enzymes, yet they're proteins. All proteins, though, regardless of whether they're enzymes or not, are made in this process of transcription, translation, to the formation of this protein. And so we're going to spend some time in the next few lectures talking about this process here of transcription in more detail, and this process here of translation, where you have these little molecular structures that are called ribosomes. They sort of look like hamburger buns, right, an upper and a lower half. So they are the ones that then bind to the RNA, and with their help, the protein is made. And exactly how that works, we'll also talk about in a future lecture. So the central dogma basically is, if somebody were to ask you what's the central dogma, it's just that information flows from DNA to RNA to protein. Okay, so we want to spend some time on DNA today, and then on next time on transcription. So little recap here on the nomenclature of nucleic acids. There's something that's a little bit confusing, if you will, but it's really not that confusing. So a nucleoside versus a nucleotide. So a nucleoside is a base and a sugar molecule. That together is called a nucleoside. If you have any phosphates added to this substance, to the nucleoside, then it becomes a nucleotide with a T. So this is a nucleotide, this is a nucleotide here, and this is a nucleotide as well. This is a nucleotide triphosphate, this is a diphosphate, and this is a monophosphate. And if you want to be really specific or really clear, then you would say this is a nucleoside monophosphate, because this is the nucleoside plus the monophosphate. This is the nucleoside plus the triphosphate. Together they are a nucleotide, right? Not really so important that, the zide versus the tide. I shouldn't say that. It's probably important. But we will mostly talk about nucleotides. What is important to know is that when they're monomers, they have these 3 phosphates, and there's a lot of energy that's stuck in these bonds, in these phosphate bonds, and they can be cut. And when they get cut here, 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 then the energy can be liberated. And that is important. We'll talk about that later on. There's also ATP that you may have heard of before. If not, we'll talk a lot about that later. They have triphosphates, and they are so high in energy because of this triphosphate bond. So what's important for you to know is that nucleotides have phosphates. Nucleoside, sugar, and base. Nucleotide has the phosphate. Okay. Let's take another look at the DNA strand that we looked at last time already. The two things that I want to add to what we discussed last time when we looked at the double strand and we looked at the base pairing and we looked at the way that thymine is always paired with an adenine and the guanine is always paired with a cytosine through hydrogen bonds, right? In between here are hydrogen bonds that hold these together. Well, when that forms a double helix, the outside of the contours of the DNA are, they're not even. They have, because of the way that the twist occurs in this double helix, there's what's called a major groove. There's more space here, and there's less space here. It's always a major groove, a minor groove, a major groove, a minor groove. And that's actually important for enzymes that bind to the outside of the DNA, and they sort of want to bind to the DNA to do something with it. For example, unwind it and look for what types of bases there are, and depending on what kinds of bases there are, specific types of proteins may bind. And they can determine that by not only the fact that these are TAs or GCs, but also whether they are producing a major groove or a minor groove. So we'll come back to that as well. The other thing that I want to point out in this slide is that DNA runs from 5' end to 3' end. That's something you'll hear over and over again, and it's important that you understand what that means. So I told you earlier that carbons get numbered in molecules, and there's rhyme and reason to how that gets done, depending on the kinds of chemicals that are used and the way that they interact with each other. That's the numbering. But what you need to know, all you need to know really at this point, is that when the numbering occurs in a nucleotide, the numbering of carbons starts here and then starts again here. This is the first set of carbon numbers, and this is the second set of carbon numbers. And because this is the second set of carbon numbers here on the sugar, in order to distinguish that from the carbon numbers that are here on the bases, these are called carbons numbers with a little prime on it. So you see that little dash there, that little comma, this little apostrophe, if you will. That's the prime. Here's a prime, here's a prime. So you will hear the 5' end or you will hear the 3' end. And what that means is just the carbon numbered in the sugar. The carbons numbered in the bases don't ever really get used for the kinds of descriptions for DNA. So it doesn't really matter what these carbon numbers are for our purposes right now. But it does what the numbers are within the sugar. So this is the 3', this is the 4', this is the 5', and then 2 and 1, of course. And so when your book talks about the fact that DNA runs from the 5' to the 3' end, what they mean is that here is the 5' end of the sugar, and that is where the next phosphate group is. And then the next phosphate group after that is bound to the 3' carbon. So the 3' carbon is here, the 5' carbon is here. And the next nucleotide will be bound at this 3' end with the 5' end of the next nucleotide. So the 5' end of this nucleotide is bound to the 3' end of this nucleotide, and so forth. Now this has a 3' end, and the 5' end of the next nucleotide gets attached to the 3' end of the previous one. And so when I say that it runs from 5 to 3, what it means is that when it's synthesized, it gets synthesized in this direction from 5 to 3. And that's what this big blue arrow actually is supposed to show you as well. So at the end of a growing nucleotide strand, you always have a hydroxyl group. And at the beginning, if you will, of the other end, you have this phosphate group. You will later see that many of the other processes that occur in the cell that require DNA always go from 5' to 3', or sometimes from 3' to 5'. And so therefore it's important for us to know how to distinguish which side we're talking about. That's the reason why you need to know what 5' versus 3' means. Okay, so then if you take the strand, and I said it's a double strand, right? But so far I've only shown you this one strand. Sometimes last lecture, only this part here. Today I just showed you this part with the bases. The second strand runs also 5' to 3'. However, it's reversed. It's in the other direction. So see how the arrow goes up in this direction while the arrow here goes from here to here? Well, that is the 5' end, and that's the 3' end. And so this strand over here runs from 5' to 3'. But this strand on the other side also runs from 5' to 3', except it runs in the other direction. And as you will notice, because of that, they also drew the sugar residues here on the head, if you will. So the oxygen is here on the bottom, here it's on the top. So same principle here, that you have the 5' end here and the phosphate end here and the hydroxyl end over here, while on the other side it's reversed, right? Phosphate group here, hydroxyl group end here. So 5' to 3' here, 5' to 3' in the other direction here. And that is called anti-parallel. So they're next to each other, they're parallel to each other, but they go in opposite directions. So one this direction, one in that direction. So that is anti-parallel. And then the last thing that you need to know for this slide is that because of that, anti-parallel, and because of the base pairing and the rule of the base pairing by which only a C with a G and a T with an A can pair, because of that, you have what's called complementarity or complementary strands. So if I gave you this strand here and I would say, well, what's the other strand? Then you could deduce that immediately from just looking at this. You would know that, oh, there has to be if there's a G here, there's got to be C on the other side. If there's an A here, there's got to be T on the other side. Can't be anything else. Can't be an A here, can't be a C here, can't be a G here. And so they're always complementary to each other. And so therefore that entire strand is the complementary strand to this strand, right? Not complementary with an I, like, oh, what a beautiful strand you are, or that's a free strand, have a strand, it's complementary. No, complementary with an E, that is meaning being the complement, being the other one that fits. That's basically what that means. Okay, so DNA is double-stranded and anti-parallel. It has 5' to 3' ends, and those are important things to know. All right, so then because of this complementarity, you have, when you unwind a piece of DNA, you always have what's called the template. You always have the instructions, essentially, of what the other strand should look like just by looking at the first strand. So you can unwind this, or you can let your cell do it, an enzyme inside your cell do it, and you can have these parental strands, as they're called. Both of them are parental strands. And then when the DNA is replicated, because a new cell is made and the entire genome is duplicated, then the new strands are called the daughter strands. And the daughter strands can easily be made from the parent strands because the parent strands can serve as the template. They can serve as the template. This is the template. The daughter is the copy. So when you have long DNA molecules, they form chromosomes. You may have heard about chromosomes in humans. Humans have 23 pairs of chromosomes. So there's one from mom, there's one from dad. We have 2 sets of chromosomes, 2 sets of long DNA molecules. And when we have 2 sets of chromosomes, that's also called being diploid. We'll talk about that quite a bit later. So haploid would be just having 1 set of chromosomes. Diploid would mean having 2. But right now, I'm only talking about chromosomes. Chromosomes are the long, really, really long, very compact strands of DNA that are basically one long stretch, except there's 23 in humans in 1 set, and we have 2, so we have 46. Dogs and cats and mice and rats and plants all have chromosomes, but they have a different number of chromosomes, so they don't all have the same number of chromosomes. Okay, and the DNA in the chromosomes, so this is the DNA in the chromosomes. This isn't quite yet all that you have when you look at a chromosome. They are wrapped around specific proteins, a specific set of proteins that has a certain set of amino acids, a certain type, number, and kind of amino acid. And it's sort of like spaghetti. If you have, very long bits, in this case the chromosomes, they're kind of hard to do anything with. And so they're so long that in order to make them a little more compact, they get wrapped around these proteins. And these proteins that the DNA is wrapped around are called histones. So imagine that you have this protein here, and then you have this big ball of DNA. And in order to make it a little more compact, a little bit more orderly, it's wrapped around these histones. There's always usually 2 rounds of DNA wrapped around one histone, and then there's another histone, there's another 2 rounds wrapped around that. That is how the DNAs sort of get held in better shape, so that the cell doesn't get lost in this ball of this wad of DNA. And that together is called chromatin. So the chromatin is the DNA plus the protein. All the proteins, there are also some other proteins that are attached to DNA that we will discuss later in the class. But DNA plus protein is called chromatin. DNA in long stretches is called chromosomes. And the proteins that the DNA is wrapped around are called the histones. And that's some more of the nomenclature that you'll need to know. Okay, so we are already at the end of today's lecture. Let me make some room here and put myself in the bottom corner. So what I want to just sum up today is that I showed you some experiments, the results of some experiments from a long time ago that showed that DNA by itself can provide a living cell with information for the new acquired functions, and that process of taking DNA and basically presenting it to a living cell, and that living cell can then take up that DNA into its own cell and use it, that is called transformation. Exactly how transformation works and why a cell with a membrane can pick up a huge long molecule like DNA, that we'll discuss as well in the future. We actually will do some in the lab, but that is the process of transformation. I talked about the central dogma, so DNA, RNA, protein. That's something that you will need to know. You should know what antiparallel means and why DNA is antiparallel, what a complementary strand is, and why that structure of a double helix that is antiparallel in nature can lead to being the template for the production of new strands, daughter strands, in during the process of replication. And that's it for today. That's all I have. Next, we will talk about transcription in the next lecture. See you then.